Hello everyone, welcome to my channel Biochemistry Concepts. This video is about the primary structure of protein. In general, the structure of protein can be classified into four levels of organization, that is primary structure, secondary, tertiary and quaternary structure. So primary structure, it tells us about the sequence of amino acids in a protein or polypeptide chain. Now coming to the significance of primary structure in terms of sequence. So each protein has a unique sequence of amino acids and that is decided by the genes. And this specific sequence of amino acids is very significant for a particular function of protein. So the function of protein depends upon the sequence of amino acids of that particular protein. Now we'll see the importance of sequence. For example, let us consider there is a protein with normal sequence: glycine, alanine, valine, leucine, methionine, phenylalanine and tyrosine. So with this sequence, this particular protein is functionally active. If the sequence is changed, now the amino acid sequence here is changed with regard to the second and third amino acid. So actually in the normal protein, the amino acid sequence is glycine, alanine and valine. But here the sequence is changed to glycine, valine and alanine. So because of this, the normal protein is converted into abnormal protein due to change in sequence. Now, there are many genetic diseases that result in the proteins with abnormal amino acid sequence. So because of this abnormal amino acid sequence, these proteins, they cannot fold properly. So that leads to improper folding of proteins, and these proteins are functionally inactive because they have abnormal amino acid sequence. So let us consider the most common condition, that is sickle cell hemoglobin, that leads to sickle cell anemia. So here the normal hemoglobin, that is HbA, in the beta chain at 6th position it has glutamic acid, but due to mutation that glutamic acid is converted into valine, means glutamic acid is replaced by valine. Because of this change in amino acid, it leads to improper folding of the polypeptide chain and that leads to the formation of sickle cell RBCs. And primary structure also determines the remaining protein structures, like it determines the secondary structure, tertiary and quaternary structures of proteins. Coming to the bonds which maintains the primary structure, the main bond is the peptide bond, also called as amide bond. So these maintain the primary structure. So when you see any polypeptide chain or protein, it has two ends. One is N-terminal and the other one is C-terminal. Left side of the polypeptide chain, we call it as N-terminal, and right side of the polypeptide chain is called as the C-terminal. So at N-terminal, it has a amino acid with a free amino group. So in a polypeptide chain, on the left side, on the left end, it has the amino acid with free amino group. So that's why it is called as the N-terminal or amino terminal. And that amino acid at the N-terminal is called as the first amino acid. In the same way, at the right side of the polypeptide chain, that is at right end, it has a free carboxyl group. So that's why it is called as the C-terminal. And the amino acid at the C-terminal is called as the last amino acid. The remaining amino acid in between the N-terminal and C-terminal, all these are joined by peptide bonds. So this is the covalent bond that maintains the primary structure of a protein.